All right. I think the microphone's working. Thanks, Rose, for that introduction. It's great to be here. Standing room only. I feel this warms my heart. Nothing like my organic chemistry class. I think you'll stay home for that one. But I'm going to go ahead, and I think I'm going to disappoint you right off the bat by saying, okay, the title of the talk is, How Did Life Originate on Earth? And the answer is, I don't know. All right? And furthermore to that, nobody knows. This is an area of active research. It is one of not only a hot area of science, but one of those questions that transcends science and gets to the deeper meaning of what is life. How did it form? It's one of these fantastic problems where we have the answer, we just don't know what the solution is. We don't know how we got there because we are the answer. The fact that there's so much life around, we know that it had to have started somehow. Now we have to figure that out. So nobody knows, but we have some ideas, and those are the things that we're going to talk about a little bit today. I think I'm going to go for about, let's say, 45 minutes, and then we'll have plenty of time for questions too. So think of questions, or I might treat it like organic chemistry. I'll call on you guys, treat you as students, so everyone should get an idea in the hopper. Okay, so there is no first we want to start off with, if we're going to talk about the origin of life, we need to know what life is, right? And it might surprise you to know that not only do we not know how life started, we don't even have a good definition for what life is, a definition that would satisfy everyone. Everyone has a problem with certain definitions of life. Some things that people typically include in their definitions, though, are things like reproduction. Life reproduces, it replicates itself. Metabolism, life will take things and eat them and convert them through chemical reactions and have cycles associated with that. Evolution, life can change over time and improve and get better, be better in terms of how it adapts to its environment, in terms of how it replicates. And also energy dissipation is something some people talk about. One of the things about life is that we exist what's called out of equilibrium. We have to input energy into living systems in order to keep all of this order that we have in our bodies or in organisms. If that energy stops coming in, if we don't eat, we die. And if we just sit there, we decompose and lose that order. You might have heard the second law of thermodynamics, where the entropy of the universe is constantly increasing. Things constantly favor states of disorder. And life is naturally ordered. So in order to maintain that, we have to have sources of energy. And people have done fascinating things like provided energy to systems that we wouldn't say are living, and they adopt order that we would kind of think of looks a lot like life. So that could be an important part of what the definition is. If you talk to NASA, so the NASA Astrobiology Institute sat a bunch of smart people in a room and said, come up with a definition for what life is. And the definition that they came up with was a self-sustaining system capable of Darwinian evolution. So got some things in there, chemical system, so networks of reactions, you could say, and then capable of Darwinian evolution. That's going to involve replicating and replication that is error prone. Your offspring don't look exactly like you. They're different. And the ones that are better are going to survive longer. Not saying that people have kids and one is better than another, but the fact that we produce offspring and all life produces offspring that's different is advantageous because of the environment, the conditions will select for the fittest of those group. In theory, that's what Darwinian evolution is. And then life gets better at surviving, reproducing, dealing with its environment. Now, you could start to poke holes in this definition because you might look at a mule, a nice happy mule there, or maybe it looks kind of sad, I don't know. But supposedly Wikipedia says that's like the finest mule in all of Argentina. But by this definition, this mule is not alive because of a mule is sterile. It can't reproduce. But I think most people would look at that mule and say that's definitely alive, right? So that's kind of like what we're working with, where people come up with definitions that seem good, but they always have some hole in them that someone's going to poke in. Is a mule alive? Is a virus alive? Think about how a virus works. A virus does not have the biochemical machinery that organisms, regular organisms, me, plants, bacteria have. But what it does have is are nucleic acids. So it will have nucleic acids that code its genome. But how a virus works is it goes around and it just finds a cell that does have biochemical machinery, and it inserts its genes into it, and then tricks the cell, essentially takes its genes and starts doing everything that the cell would do with its own genes. So it kind of, viruses hijack living, other cells, proper organisms. So is the virus alive? Can it sustain itself? Well, it can't sustain itself if it isn't in the presence of an organism for which it could be a parasite. So again, those definitions kind of break down, and a lot of people would say viruses aren't alive, but I think most people in my field would say viruses are alive. So, there's a lot of gray in what we have to deal with. My favorite, though, to kind of wrap up definitions of life, my favorite treatment of this was a paper not by a scientist but by a philosopher, Carol Cleland, who said maybe we should think about life how someone in the 1700s would think about water. So back then, you can imagine if you were trying to come up with a definition for water, you might have problems. Yeah, that looks like water, and it is, but this is also a form of water. So this is a cup of tea. I mean, we know that it has mostly water in it, but the question is, what makes tea? Is tea water? Is water, what's the difference with that? We could also have salt water. This is the Dead Sea. So water can have colors. Water can have different tastes. Water can have, different physical properties, like its boiling point, its freezing point will change based on all of these things will have different values for that. It wasn't until science progressed, and then Cavendish, Lavoisier, James Watt came around and said, what really makes water water is on the atomic level. It's H2O. It's one atom of oxygen bonded to two hydrogens. And all of these other manifestations that you could say are water, salt water, is actually, those other properties are imparted by the impurities in your H2O, whether they be salt or tea or dyes, whatever. So those are the other properties that are those are the other substances that are imparting those properties on water. But water itself, pure water, is boils at 100 degrees Celsius, freezes at 0, is clear, doesn't have an odor, and we could really understand what's going on. So maybe but if you went to someone in the 1700s, if you went to George Washington and said, come up with a definition for water, he'd have a lot of problems coming up with one solid definition that you couldn't poke holes into. Now we obviously know, and anyone could rattle off H2O. It's become that ingrained in our knowledge base. But perhaps we're at a point where what water was in the 1700s, life is for us now. We kind of know what's going on, but we don't have that one solid definition that really encapsulates life. And there might be something fundamental there that scientists need to keep their eyes out for to develop that one hardy definition. And coming up with solid definitions about things I always love pointing to this is Potter Stewart, who is an Associate Justice of the Supreme Court. And in a case in 1964, he came up with this passage of, I shall not attempt to define the kinds of material I understand to be embraced within that shorthand description, and perhaps I could never succeed in intelligibly doing so, but I know it when I see it. Does anyone know what he was referring to when he made this ruling? Pornography, specifically hardcore pornography. He couldn't come up with a definition for it, but he knew it when he saw it. And I think that a lot of us could, while we might struggle to come up with a definition, we know life when we see it. And some of those things include, like when you really drill down to the molecular level, things like a nucleic acid genome. We know that organisms have DNA and RNA, and those are the molecules that code the information that's important for the rest of our biological machinery. All organisms share that. We have ribosomes, which is where the genomes that are encoded in our DNA are used to, that information is used to synthesize proteins from amino acid building blocks in a particular order. And the order of amino acids that go into those proteins is what controls their chemical properties. All organisms share that. You might have heard the molecule ATP, which we use as an energy currency. So when you eat stuff, your body ends up producing ATP, and that ATP is essentially the common currency that cells use to power what's going on inside them. All life as we know it uses that. Membranes, keeping cells enclosed and keeping organelles, smaller pieces that are inside those cells enclosed, use molecules that look like this, that assemble kind of into these structures that kind of divide the house into rooms, or even keep, or kind of like the walls of the house. All life uses water as a solvent. So when we want chemistry to go on, we have to have some liquid medium for that to happen. For life on Earth, that's water. Water is extraordinarily important for life as we know it. Not to say that all life necessarily in the universe would need that, but life here does. And then I put something, ion gradients here are another thing. We all, ions are charged atoms, essentially, or molecules, and your body will separate those inside or outside the cell and spend a lot of energy pumping things in and pumping things out. And those are universal to life as we know it. These are not all the features that are universal to life as we know it, but they're some of them. So we kind of know what we're looking for. And as a chemist, these are important things because we want to look at properties that all life shares. And if every organism that we know of has that system, then that system either evolved really, really early, or it was present when life originated. Right? So those are the things that make the most sense to look at. It wouldn't make sense to look at eyeballs as something important to life, because of not every living thing has eyes. So we know that it's not critical to life as we know it to have that system. So we kind of focus on the things that every organism has. Okay, so drilling down, what I'm interested in as a chemist is how did an abiotic mixture of chemicals, so abiotic, non-living, so just molecules, whatever was there when the planet formed, how did that mixture develop into an auto-amplifying network of chemical reactions? So simply put is biology, as what are cells? Cells are sacks of organic reactions that are taking place. How did that network of thousands of reactions that take place in your cells, how did that start? Did it start as smaller pieces? What smaller pieces? What molecules were there in the beginning? How do you get into a system that is auto-amplifying, which is essentially another way of saying it reproduces? In chemistry, we really don't think of molecules reproducing. Reactions happen and they have products. They take starting materials, they convert them into products. But how do we make something that makes products, that has another product, that has another product, that has another product, that eventually feeds back into making the starting material for that first reaction that you had, so that you could complete the circle and the system will just take off and keep on reproducing more and more of itself. Biggest mystery, in my opinion, in the history of the planet, maybe you could say in the history of the universe, the Big Bang would probably give it a run for its money. But in the history of the planet, I think life is the biggest thing that's happened to it. And one of the greatest questions of all time. And what's nice about it too, it's an interdisciplinary field. It's one where practically every branch of science has something to contribute. We look to astronomers who observe moons in our solar system. They observe other planets in our solar system to try to find clues for, is life there? How would we know if life happened on Mars? What are we looking for? Or on other planets. We send probes there that have instruments on them to kind of measure things, but we have to design those instruments with kind of these goals in mind of what we're looking for. So what should we look for? And what we look for on Mars might not be the same thing that we look for on one of Saturn's moons or one of Jupiter's moons, which have different mixtures of chemicals and different conditions on them. Astronomers have also gotten great at looking not only at planets in our solar system, but what are called exoplanets, which are planets that are on distant stars. So there's a telescope called Kepler, which just died. It just ran out of fuel. But it has discovered, using the data from that telescope, scientists in the past 20 years have discovered hundreds of planets around distant stars. So we know that there are all sorts of stars. We could see stars because they emit light, and there are, billions and billions of them, as Carl Sagan would say. But each of those stars appears that it has multiple planets around it. Now, those are really hard to detect because they're not as big as the star and they don't emit light. But that telescope was good at detecting the wobbles in the light that came off those stars as planets went around them. And if we got lucky, and with so many stars out there, we can get lucky a lot, if a planet happens to go between that star and us, we could actually see a dip in the light that's emitted from that star and attribute that to a planet that's crossed it. And then even better than that is, we could see what wavelengths of light go down relative to others. So white light is composed of a spectrum of all sorts of different wavelengths, all the colors of white light, plus extending into the UV and IR. So we could actually see what the atmosphere of that planet absorbs. And that's really interesting because we really would love to find a planet that has oxygen in its atmosphere, because the way that oxygen got here on Earth was that life made it. Photosynthesis was responsible for making it, so it might be a key, oxygen, an atmosphere of oxygen might be a key biosignature for life, and we could detect it in planets that are light years away. One last thing that astronomers are good for, astronomers are good for many things, but personally, what I'm interested in they're doing is, it's pretty amazing. We have landed probes onto comets. So comets are moving around the solar system, they're orbiting the sun. We've put a probe down on a comet and had it hammer into it and make a plume of dust, and then we take a spectrum of that dust that comes off to determine the molecules that are in it. And so as a chemist who wants to know what ingredients I have to go back to the prebiotic kitchen and cook, these comets are fascinating because they are essentially snapshots of what our solar system looked like when it initially formed. Our solar system is not what's called a primary solar system. It's a secondary or later. We did not form at the beginning of the universe. We formed from the contents of destroyed solar systems and destroyed stars that went nova previously, and then kind of the material recoalesced into our solar system. Now, we really don't know what that looked like by looking at what's here on Earth, because of the molecules on Earth have been affected by life. They've been affected by all sorts of things that happen on the planet, tectonic issues, things like that. But what's nice about comets and asteroids is that they did not hit planets in the beginning. They did not accrete into the planets or moons that are around. And they have not been affected by life. Life, as far as we know, has not originated on a comet or an asteroid. So we could kind of look and see what molecules are on these comets and asteroids and think that those same molecules would have been what rained down onto the Earth when the Earth was forming 4.5 billion years ago. So it's kind of like getting a picture of what the solar system might have looked like 4.5, 4.6 billion years ago when it was just forming, whereas all that information has been lost on Earth. So that's really useful information. Geologists, they look at Earth and they can dig up things and date them using radioactivity of different isotopes. We can date things that are old, and things have been isolated that date back to, I think, 4.3 billion years ago, despite the fact that that the planet continually chews itself up and respits itself out. It's really hard to kind of find things that date that old, but we have found things, and we get clues from that. Like, someone has found minerals that were in Australia that suggest that there was an ocean present, that water was present in about at least 4.3 or 4.4 billion years ago, when these minerals date to, because we know that these sorts of minerals only form in the presence of water. And in addition to that, some minerals, we know what their structure looks like when they form in water at different temperatures. So we can kind of tell that the Earth was probably warmer, at least when these minerals formed. So there's all sorts of information that we can get out from these old samples. Chemists, the heroes, we're going to be the ones who figure out what exactly went on. We take the information of what was there, what molecules were there, and what conditions were there, temperature, lightning, how volcanic activity, what sorts of environments we had on the Earth back then, and we figure out what kind of chemistry would have gone on. And chemistry, it's called the central science. It's between the physics end, the astronomy, and biology. So I always like to say, physicists and astronomers, what they do is too abstract to get anywhere on this problem. And by the time you get to biology, the problem's already solved. So chemistry's in the middle. We're the ones who have to figure things out. Biologists are also extraordinarily useful while they look at things that are already living, so they're by definition past the origin of life. They do all sorts of cool things like look for minimal life. What's the least number of things that you need in your cell in order for it to reproduce and live? They will take genes and they will knock them out one by one, and then see if their injured organisms can still reproduce, and see how many things they could turn off before life can't function anymore. And that's useful information. What are the critical properties of life that we really need, and what things don't we need so much? And then also, looking at biomolecules that we know are important, nucleic acids, proteins, and seeing just what those biomolecules are capable of. So it's a team effort. The field is very interdisciplinary, and everyone has something to contribute. Okay, so here is a good timeline of the geologic history of the Earth that I borrowed. And we are interested in what's going on over here in the Hadean. So I always like to point the example to, if the Earth's history were a 24-hour clock, and the planet formed at midnight, and where we are now is midnight the next day, life formed by 4 a.m. is pretty quick. Didn't take too long. Granted, we're talking about millions of years, but by 4 a.m. life had formed. It took from 4 a.m. all the way to 9 p.m., so at night, to go from single-celled life to multi-celled life. And in this time scale, humans don't come into the picture until 11:59 and 30 seconds or so. So we are the products of evolution. But life was there. It was a lot easier for some reason to go from nothing to single cells than to go from single-celled life to multi-celled life. And some important things that happened. So 4.6, the solar system formed. By 4.5, the Earth, enough material had coalesced to form the Earth. We have rocks that date back, these Jack Hill's zircons, to I think 4.3 or so billion years ago. There are important things like rocks where it looks like there are microfossils in them have been found that date back to 3.5, 3.7, and more recently 4.2 or 4.3. And the way that these microstructures in these rocks look is that we believe that they could be evidence of microorganisms, and that they're fossilized microorganisms that suggest that life originated somewhere in here. The ones at 3.5 are a lot more robust. People think that those are pretty much biofossils. The ones that are at 3.7, there's a lot of concern over whether those just might look like they could have been living but were naturally formed. And there's even a heightened sense of that for the more recent data that's at 4.2, 4.3 billion years ago. But life originated pretty darn quickly in the grand scale of things. So there might be hope for us figuring, if something happens that quickly, there might be hope of us figuring out, how this could have happened, because it didn't take a whole lot of time to take off. All right, want to talk about the setting for what's going on. So it's on Earth, but it doesn't look anything like the Earth that we know. We would not recognize the planet that life originated on. Again, we're talking, let's say, 4 billion years ago, to have a nice round number. The Earth was probably hotter. The evidence for those zircons that were isolated suggests that, they're like minerals that are like opals that formed, and suggests that at least where these things formed, that date back to 4.2 or 4.1 billion years ago, that the planet, the water where they were was 70 degrees Celsius. So that's pretty darn warm. Not something that we'd want to swim in. The atmosphere, we think we have problems with CO2 now, and it's 0.04% of our atmosphere. 0.04. The atmosphere of CO2 back then was very, very high in carbon dioxide, and we're talking like possibly greater than 90%. And this is one of those things where we could look at our neighboring planets. So Venus and Mars are either side of us. Both of their atmospheres are over 90% CO2. They have different densities than ours and than each other, but they all have high CO2 atmospheres. So that's probably what we were dealing with back then. And again, not a very hospitable place for us. You would feel, as you breathe in that air, you would feel the kind of that same stinging feeling that when you get Coke in your nose when you laugh so hard. Like it's not pleasant. Nitrogen was probably the rest of the atmosphere. Water vapor was in there. There was no oxygen. Like the upper estimates of how much there was probably about 10 to the minus 15th times what we have today. So essentially anoxic. And that has consequences. If there's no oxygen, there's no ozone layer. And if there's no ozone layer, then that means all that high energy UV radiation from the sun is penetrating to the surface. That can be destructive to organic molecules, but also it can be a source of energy that reactions can use to take place. So it's just something to keep in mind. It's not necessarily good or bad, it's just what it was. Back then too, as the planet was hadn't gone through all the tectonic activity that we have today, there would have been less landmass. So probably it might have only been as much as 5% land. The surface of the planet back then, it would have been covered with an ocean, much more. So that presents a problem, right? Because if you have a huge ocean, that's going to present problems with diluting your cells. If you've got a cell that's developing, its contents have to be sequestered from the rest of its surroundings. That's one thing that makes life, is that you could tell where life begins and ends. It has a membrane, something encloses you. We have skin. If you just have a puddle of reactions that are developing and it gets washed away in the ocean, all your molecules are going to get lost and diluted. So that presents a huge problem. It's something called the compartmentalization problem that we have to deal with. What did the first cells look like? How did you separate things? The ocean was probably more acidic than it is now, with all that CO2 in the atmosphere, and also with dissolved metals like irons that were present back then. But there are some things that life needs now that we wouldn't have had large quantities of, like phosphates. So if you're into lawn care, you know that one of the important fertilizers are levels. One of the three key indicators that you get in the fertilizer you buy are phosphates. Living things need phosphates to grow because they form the backbone of our DNA and RNA. If we didn't have phosphates, how do you form those molecules? So the oceans back then would not have had high levels of phosphates, so that presents a real problem to how life as we know it could develop. The Earth was also violent. Because we were early in our solar system and there were a lot more comets and asteroids, the chance of an asteroid or comet hitting the planet was much, much higher. So these things were whipping in all the time. And you might remember that an asteroid is what was responsible for mass extinction events like the extinction that killed the dinosaurs. Those things would have been happening with increased regularity back in the time that we're talking about. So that presents a problem to things that are developing, is that they could be destroyed by those events. But it also presents an opportunity because of those are high energy events where maybe those shocking impact events could have synthesized molecules as well. So again, give and take, but that's what life back then would have had to deal with. Lots more volcanic activity too, and all the things that volcanoes spew out of themselves would have been present in the atmosphere and around, and that would have led to also increased electrical storm activity, things like that. So the Earth back then looked, I mean, I'm not saying that it's this, but it doesn't look like what we have now. There's no idyllic green pastures and bright sunny days. Or maybe there are bright sunny days, but there's ionizing UV radiation raining down on you, which isn't so happy. There we go. Okay, so some problems that we're looking for. Early genetics. So we now have DNA and RNA to store information. Those are really complex molecules. How could we store information in more simple ways? Like what might have been present to store information before these molecules came online? Catalysts. Catalysts are species that lower the energy, the activation energy required for reactions. They speed chemical reactions to get them to go in a time frame that is more convenient. We have proteins that are enzymes that do this for us. But enzymes are huge molecules that are very complex and also in many ways delicate. They could break down. What catalysts were important to the development of life before proteins kind of came online? Self-replication. How can you make anything that makes more of itself? That is a really challenging thing to do on a chemical level. All the great self-replicators in the world, for the most part, are biological. But before you had biology, what's the chemistry that could have done that? One interesting thing that we didn't talk about when we talked about definitions of life is fire alive? Fire reproduces. Fire takes fuels and converts them, uses that energy. Would people say fire is alive? By some definitions, maybe. I think most people would say no, but again, something to consider. Energy, what sources of energy were important? UV light, or any photons from the sun. Radioactivity could have been important. Tides would have been bigger because the moon was closer. Could you use that energy, so essentially gravitational energy? All sorts of chemical processes, redox processes, reduction oxidation reactions where electrons flow, those would have been important in theory. And we talked about the compartmentalization problem. What were the first cells? How did life separate itself to protect itself from the environment that was around it? So these are all kind of like the top level things that we look at in the field. So let's talk about some history and what progress people have made. So back 200 years ago, one of the prevalent theories about living things versus non-living things is that there was what was called vital force. That molecules that were associated with life had this force, and living things could only be made, these molecules could only be made from living things. There was something, they didn't know what it was, but there had to be this vital force there to get these molecules made. Inorganic materials were not made by living things, and they had other ways of being made that didn't require this vital force. So one of the first experiments or results, I wouldn't say it was an experiment, that kind of upset this idea was this synthesis of urea by Frederick Wohler. it was known, urea was known before this reaction because of it was isolated from urine. And so this was one of those molecules that was assumed to have that vital force within it. This molecule that was started out, this ammonium cyanate, was known as, or treated as an inorganic chemical, one that was not associated with life. Well, Wohler was playing around trying to do something else and heating and cooling this, and then noticed that he produced urea in this process. So this kind of shook the world, the scientific world, on its head by saying, this is the first example of something inorganic, or assumed to be inorganic, being converted into something organic. And so he phoned one of his friends who was in the field and said, hey, I got to tell you, I made urea without the use of kidneys. They made urea at the time by isolating it from human urine or dog urine. And he was able to make it in large quantities without the use of a living system. So this was one of the pioneering experiments of the origin of life field, because of it was assumed by many before that there was something about life that you could not originate it from scratch. Life had to be there. So they didn't know, life just always was, and life is responsible for making these molecules that are associated with biology. But this kind of said, no, there are paths to go from inorganic materials not associated with living things that would have been present on the earth when it formed, to forming life. So life could essentially develop from chemical evolution, reactions evolving until they became biology. And so this was a process that took decades to kind of quiet the people that were proponents of vitalism. This sort of idea didn't really, the nail in the coffin was kind of more towards Louis Pasteur's time, later in the 1800s, where he ran experiments that we might be more familiar with about spontaneous generation, where people thought that life could form out of nothing. And he ran carefully controlled experiments that showed that if you separate the food or the material, the media that life could grow in, and keep it sterile and not exposed to get contaminated, that life will not originate, and that life, the spontaneous generation of life that people were seeing and attributing to spontaneous generation, was actually just contamination from living things that happened to get into the food sources that they were growing in, whether it be maggots developing on meat. You could leave out meat and then come back and find maggots in it and think that those maggots just developed out of the meat that was there. But if you put the meat in a jar and put cheesecloth over the top so that flies can't come in and insert larvae, then eggs that develop into larvae, then you don't get maggots. Same thing with bacterial like broth that bacteria could grow in. If you just make it and leave it out so that bacteria from the air can land in it, it starts to grow in that soup. But if you put it into a flask that has a curved edge that bacteria can't fly into, then that nothing grows in it. So it took a while. It took most of the 1800s to kind of put an end to the idea of spontaneous generation, and then also this vital force element. So 100 years later, or so, after Louis Pasteur, 1953, a very important experiment was done where this person, Stanley Miller, who is in the laboratory of a Nobel laureate, Harold Urey, set up this glass apparatus in a really just kind of fundamental, neat experiment. It was assumed that the atmosphere, remember how we talked about how it was mostly CO2? It was assumed that the early atmosphere might have been made of these reduced gases: water, methane, ammonia, hydrogen. And what Miller did was to make this apparatus, put these gases inside of it, these small gases that would have potentially been in the early atmosphere, and then take an electric current and arc it through this mixture, kind of to simulate lightning, and to let this go. And as this proceeded and he kind of had this condenser down there so it could go into water and then get recycled and come up. As this reaction proceeded, the water became brown and then it turned darker. It was obvious that chemical reactions were occurring. And after you let it go for so long, days, you actually could go in there, fish out, run an analysis of the chemicals that were there. And while you only started with these simple gases, you ended up with all sorts of chemicals in there, many of which are things that we associate with life. Glycine, alanine, lactic acid. Glycine and alanine are amino acids. We talked about how amino acids are the building blocks for proteins. Proteins which are enzymes which are responsible for getting our biochemical machinery to run. So this simple experiment, which simulated the early atmosphere and a simple source of energy like lightning, kind of showed that you could make the simple building blocks which could, in theory, assemble into these important biomolecules that we know today. So really fantastically, this kind of set the field on fire, showing that what was possible with very simple experiments to generate all sorts of complexity. And then when a chemist looks at that list, the complexity kind of takes off. I guess what I would equate it to is, imagine that you're at the grocery store and you just walk through an empty aisle. That's kind of where we were in 1952. Maybe there was like when there's a snowstorm coming, there's like there's no bread or toilet paper or anything like that. There's just like maybe a old pack of like frosted shredded wheat or something like that that's not very appetizing, whatever. This is like, as soon as this experiment happened, it's like walking into a supermarket and saying, all right, I got flour, I got eggs, I got sugar. We can make a lot of more complex molecules from these smaller ingredients. So it was a really exciting time. And then there's this idea of, you might have heard prebiotic soup or primordial soup, which is that chemical reactions could take place with the molecules that were there to develop more and increasingly more complex molecules, and molecules that would have been potentially more important to life. And there's work that's going on internationally. John Sutherland is a person who's in the UK who does a lot of organic synthesis, and this is a figure that I had that I made that was associated, that kind of explains some of his work, is that you can take the molecules that we saw in the previous image and some other simple ones and start to make things that look like the building blocks for nucleic acids, DNA and RNA, that look like the building blocks for proteins, the amino acids that are there, that are the building blocks for the lipids that make our membranes, that are the building blocks for carbohydrates that are our sugars. There's all sorts of interesting chemistry that could go on, and these molecules could, in theory, build up to the point where life could start, or systems could start picking and choosing things that to kind of evolve into a system that has more complexity and function. So we really are at the point where we've shown that it's possible to make a number of molecules that are familiar to us. Now, that doesn't always mean that just because you have the pieces that they could assemble into the machine. You can imagine going to an auto parts store, buying some tires, buying a steering wheel, buying bolts, and just throwing it in a pile. That doesn't make a car, right? So we have the pieces, but that we don't know how to make the machine yet. That's kind of one of the places where we're stuck, but one of the places where we're looking. One theory that people have, like, we always like to look at environments that might have been hospitable to life, or at least the development of life. And these, we don't really encounter them in our everyday lives, but if you go to the bottom of the ocean, especially where ridges are, that there are these formations called HyperThermal vents, or deep smokers. What they are is they kind of emit, they cycle in water, and then they will emit minerals in those at very high temperatures. And those minerals that are in them could serve as catalysts, they could serve as feedstocks for the synthesis of larger organic molecules. There's all sorts of electrochemistry, which is possible, that kind of suggests that there are processes with energy that might have been important. Like we talked about how life needs energy. There's all sorts of ways to harvest the energy of these molecules. And so vents have attracted a lot of interest as places where cycles of chemical reactions could develop. So they're very exotic locations. We're at the bottom of the ocean, so the water around here is like 1 degree Celsius, 2 degrees Celsius, but the stuff that's being pumped out very close are hundreds of degrees of Celsius. The water could be hotter than 100 degrees Celsius, hotter than the boiling point at the surface, because it's so far down and the pressure is so high. So there's all sorts of thermal energy there, chemical energy there that can get things to go. So another place in the field that people are really excited about is what happens at these vents. Could life have developed here? But nothing so far that's anywhere close to anything that looks like an organism. Just simple chemicals reacting in various cycles that kind of suggest that energy can be harvested, that complexity can be developed. Another fascinating theory, I just kind of want to run through some of like the areas that people are looking at, is a theory that was developed by this man, Graham Carn Smith, about a clay hypothesis. What if life, when it started, didn't look like life as we know it now, but was based on rocks and crystal formation? Because of one thing's, if you've ever kind of grown crystals of sugar, like to make rock candy, you know that crystals template the formation of more crystals. If you throw salt into a saturated solution of salt water, you could start to see the formation of more crystals templated on the seeds of crystals that you added. And so crystals can kind of template their own formation, and that's what life does in many ways, that DNA and RNA template their own formation when you're copying them. So the idea with this theory is that maybe life initially was, instead of carbon life, it might have been silicon life. And those patterns of molecules that form those crystals templated their own formation. You make more crystals, but then eventually over time small molecules took over, and that eventually got those molecules copying, and then they kind of shed off the rocks altogether once they had the machinery to do the copying themselves. And then another popular idea of, and perhaps the area that's the hottest in origin life research, is what's known as the RNA world. So we have DNA and RNA, and if you're familiar with high school biology, you might remember that DNA kind of is good for long-term storage of information. It gets transcribed into RNA. RNA then goes to the ribosome, and it's what is translated, the information, the RNA that came from the DNA, is translated to code which amino acids go together in which order to make proteins, and the proteins are the molecules that are responsible for doing the chemistry. So DNA holds the information, it gets transcribed into RNA. That information then is used to code the formation of proteins, and proteins are what do most of the work for biochemistry. So that is what's called the central dogma of biology. But there's something interesting. RNA, which is in the middle, can also catalyze reactions. RNA, not all RNA can do this, but RNAs that can do this, that can kind of, proteins are really good at folding up into three-dimensional structures, which is why they're so good at catalyzing things. RNA can also fold into some structures like that. So RNA can kind of do both. RNA can both store information and it can catalyze reactions. So the idea of the RNA world is maybe there was a time before we have DNA kind of as that developed to be really good at storing information, and proteins really good at catalyzing things, that RNA did both. And there is good solid evidence for this idea, because if you look at ribosomes, so this is a ribosome, ribosomes, which are responsible for making proteins in your body, in your cells, the active sites where this happens are all composed out of RNA, out of nucleic acids, the actual active sites. There are proteins that are all around it, but where the actual protein synthesis goes on, that is all catalyzed by RNA. So the fact that the active site of ribosomes is made out of RNA suggests that RNA might have been responsible for doing everything before proteins and DNA came online, evolved to come online. So a lot of people are looking at just what can RNA do, and also how do we make RNA. RNA is actually, it's complex, it's a big molecule, they're big biopolymers, they're hard to make, and they fall apart in water a lot faster than you would imagine they'd be made if you don't have life around to kind of make them. So there's a big question as to how, well, okay, RNA, it looks like it was really important at some point, but how did you get to that point? So it's kind of like building a bridge from two sides of a riverbank. What molecules were there? How far can you go in terms of developing complexity? Life is over here. How far back can we go? We're kind of going back to where RNA was, but how do we build that middle section of the bridge? That's where we're stumped at the moment, and we are trying different things to get there. Okay, so I want to kind of talk about, that's kind of like an overview of the field, what exactly we do in our lab in the next 5 or 10 minutes. We look at, again, going back to systems that all life has. We want to focus on those because if they were either evolved really early or were there at the origins itself, they are good handles to kind of get involved in to try to figure out what's going on. So we like to call these biochemical fossils. They have persisted in the living record of life as something that were probably there from life's origins and that now every organism has. So the first thing that we're interested in our lab is potassium. Every organism on Earth enriches potassium. You spend about 1/4 to 1/3 of your energy in ATP pumping potassium into your cell and sodium out. That's why we eat our bananas. This is why when we fertilize our grass, potassium is important. And if you put sodium chloride, salt, table salt on your grass, it kills it because of life prefers potassium inside cells to get chemistry to work. We don't have a periodic table here, but if you were to look at sodium and potassium, they're right next to each other on top of each other. And they had the exact same chemistry from an organic chemistry standpoint in that they really don't do any interesting chemistry. But if they're so similar, why does life insist on having one and not the other? And why does it spend so much energy maintaining that, suggesting that it's really important? That's one thing that my lab is focused on looking at. And so what we do is we run simple organic reactions like reactions of peptides. Here they're hydrolyzing, so this is a destructive reaction. And we basically wanted to see whether those reactions change or are different in the presence of sodium versus potassium. And it turns out that, so what we do is we use a technique called NMR spectroscopy, where we could look at the hydrogens that are in these molecules. They give off different signals, and we could use these signals to monitor these reactions as a function of time. That's going to let us know how fast this reaction goes, and we could measure what are called kinetics and a rate constant for that, and we could see how the presence of sodium or potassium affects the rates of these reactions. And so that's what we do over on the main campus of SLU on a daily basis. And the thing that we were interested to find, which was completely unexpected, is that these reactions have different rates in the presence of sodium and potassium. And so here we're looking at a hydrolysis, a destructive reaction of peptides. So it's breaking up essentially the things that form proteins into their building blocks. And we find that the destruction of these linear peptides occurs much faster in the presence of sodium, whereas the destruction of these cyclic peptides, which don't look like things that we have in life for the most part, occurs much faster in the presence of potassium. And we got excited about this because of more of the same there, is that in theory life wants to take the building blocks, the amino acids, and go and build longer and longer polymers that eventually get into big proteins, which then fold up into 3D structures that can catalyze reactions. So we want to go this way. We find out that If you're in the presence of potassium, the rate of hydrolysis going in the wrong direction is slower, and that's what we want. Potassium favors getting longer and longer and longer, whereas sodium relatively kind of favors breaking things apart. So potassium helps you in that manner. What's also interesting is that these cyclic peptides are kind of like wastes of amino acids because they can't get longer than these two, just two things bonded together. So if we had reactions which sped the hydrolysis of this to get back in the productive chain, we would want those. And we find that the potassium speeds the breakdown of this into something that starts to get back in this chain. So we're kind of excited about these results in terms of the implications that they have for why cells might want potassium in terms of favoring the formation of longer and longer peptides, which can then go in to form proteins. So that's one major aspect of the lab there. What we're focused on now is seeing how potassium and sodium affect the formation of longer and longer peptides. So we can make, this is glycine, which is the simplest amino acid. It's the one that's produced in the highest quantity during that Urey-Miller synthesis. And if we heat up glycine and then we cool it down and re-wet it and then cycle this reaction, we can get longer and longer peptides and detect them by this what's called high-performance liquid chromatography. We have an instrument which separates these oligomers, which is just like different oligomers every time you add an amino acid. And we could kind of measure the synthesis of longer and longer peptides based on this, these conditions. And so what we're kind of looking at now is the importance of how potassium favors holding on to solvents. So you might have, if you ever have put down like calcium chloride, like those white pellets when winter's here and you're trying to de-ice your sidewalk, you might have noticed that if you ever spilled one in your car or spilled one in your house, that you had those white pellets, but you came back an hour later and it was just a puddle of water. And it's not because of someone spilled something wet there, it's because of those salts are really good at picking up moisture from the atmosphere to the point where they pick up so much moisture that they dissolve in it and form a puddle. And what we found, and this is a process called deliquescence, and what's interesting we've looked at is that if you take potassium salts of different anions, so what the potassium is paired with, so these are all potassium and these are all sodium, and you expose it to an atmosphere of constant temperature and humidity, a lot of the potassium salts pick up moisture to the point where they dissolve, like all these black things, whereas a lot of the sodium salts just stay dry. They don't sequester moisture. And what's interesting is when you look at the anions that are, the potassium does this, carboxylates and phosphates, these are the anions found in DNA and RNA, these are the anions found on proteins. So a lot of biomolecules have these anions that are deliquescent for potassium. So we're thinking that potassium might have also played an important role in how early cells might have retained moisture. And remember, we talked about all life uses water, right? Cells need to stay hydrated to get these reactions to have a chance of going. Because of undissolved things, reactions are very, very slow. So it's kind of interesting that potassium salts support that retention of moisture, whereas sodium salts do not. So maybe this is the reason why cells need potassium. There's also, on a different grant, a project going on in my lab which kind of just started last year. We are not only interested in how life developed on Earth, but what about other places? Maybe places that aren't covered in water, but are covered in oil. Saturn has a moon called Titan, one of its many moons. And what's interesting about Titan is it's not covered in water. It's too cold for water. There's ice there, but it's not a liquid. And so chemical reactions in there to go slowly. But there are lakes and rivers on Titan. They're just made of methane and ethane. So essentially very small oil. Kind of like the liquid that's in a lighter. Like when you shake it up and you kind of see that, except a couple of carbons shorter. So what we are trying in our lab now is to figure out how would the rules of the game change if life weren't to develop in water on a planet, but were to develop in an oil solvent. So it's something that looked more like gasoline than water. And you know that if you've ever had salad dressing that you had to shake up that separated into oil and water layers, that those two liquids have very different properties. And what we're kind of envisioning in this project is looking and seeing, say something like a comet that carries a lot of moisture on it were to hit a planet like this, and you had systems that were both had liquid water and liquid hydrocarbons, liquid oils on it. What special chemistry could happen? What would those conditions kind of favor? What kinds of reactions would they favor, and how do the rules of the game change when you're in a system that has oil versus when you're in one that doesn't? And so a little whoa. Back here, one thing that we're interested in is kind of showing that peptide formation, kind of stringing together amino acids, we're finding goes a lot better when you have those two phases than when you do reactions in one or the other. So here we're coupling amino acids into longer and longer peptides, and we're finding that if you run this reaction in a biphasic system, you get longer oligomers, so longer chain molecules. This is what's in the organic layer, this is in the aqueous layer. But if you run the reaction in just the oil alone or just the water alone, you don't see the same yields, they're lower, and you don't see the same lengths, they're shorter. So there's something that's beneficial about having the molecules being able to partition between both of those layers that there isn't if you just have one or the other alone. So kind of early work from our lab that we're just kind of getting started, but a preview of that. I don't think any tadpoles are going to jump out of our flask anytime soon, but this is the kind of stuff. These little peaks here get me very excited. It warms the cockles of my heart to see students bring data like that. Anyway, that's just to close up. Here's some, if you're interested, we're in a library, so I figured I'd recommend some books. Here's my favorite book, Origins, which is by a guy, Robert Shapiro, who can only be called a curmudgeon. And he likes to point out all the holes in people's ideas, the ones that we talked about today. So if you're interested in a skeptic's guide to kind of what's going on, I really recommend this. You could find these books on eBay for like $2. They're secondhand and very cheap. This one's well worn. Blueprint for a Cell, if you're more of a biological person, is another kind of neat little book that I think is interesting relative to the origin of life, kind of written from a biologist perspective. My email address is here, paul.bracher@slu.edu. Happy to answer questions there too. Want to acknowledge all the students in my lab who actually do the work that goes on and that I kind of talked about here towards the end. We've had projects funded by NASA, by the NSF. We are one of the nodes of the current NSF Center for Chemical Evolution at SLU. This center is an NSF-funded center that's headquartered at Georgia Tech, but SLU is a major node of that center. Here was our annual meeting from earlier this year. There are nodes all across the country that are kind of dedicated to different aspects of the origin of life problem from a chemical perspective. And one of the major nodes is right here in St. Louis. Headed up by this. We have meetings, a couple of meetings every week, and this is kind of the major funder of our research right now. And I love this figure that the center kind of developed. This is actually a mural that's been painted in a high school I'm not sure if it's high school, but a school in the Atlanta area that was designed by two of the graduate students who work in the center, that has all these little kind of pieces of the origin of life puzzle just put together in kind of this beautiful thing. And the final thing I'll say is that I have this mural we're in a library, so on bookmarks that have been produced by the center. So if you'd like a bookmark, come up here. I think I have plenty, 50 or so. And then if you happen to be in college, they gave me this little flyer for their undergraduate research opportunities in Georgia, if you want to head down there. If you're a college student, if you're in high school or middle school, you will eventually be in college, and there'll be opportunities like this down the road. So that's all I have to say. Thanks. And happy to answer questions for as long as you want. Question?
Craig Vitter seems to be a pioneer of the Human Genome Project, who was responsible for a lot of the private end of that, and sequencing machines that sped it up dramatically. He was working about 10 years ago, as I recall, on a synthesis of life project in the lab.
Yes.
And he claimed that he was really close. Whatever happened with that project?
I'm not sure what they. So there's Craig Venter, who was the pioneer of the Human Genome Project. He is actively involved with a number of other people in what's called synthetic life or synthetic biology, and basically building a cell from scratch or from the pieces. I am not sure exactly where they sit. They've sat there. I know that they've made a number of milestone achievements with that, in terms of synthesizing molecules in the lab, and as opposed to having biology make them. I'm not a biologist, and I don't want to do them a disservice, but there's nothing that they haven't solved the problem yet, and there are people actively trying to put organelles together and things like that. One thing that does come to mind that was really interesting is, we didn't talk about it today, but all life has the same, the molecules in life have a handedness, kind of like how you have a glove, where this is a right-handed glove. If I put it on my left hand, it doesn't fit, right? We can look at life, and all the molecules in life are right-handed. So if you put like a left glove or a mirror image molecule, like these gloves are mirror images of each other, the machinery doesn't work. Just like when you put your left hand in a right glove, it doesn't quite work. Well, someone recently synthesized an enzyme that's the mirror image enzyme. So all of the amino acids, the thousands of them are in that, are all the opposite, the non-natural configuration. And it did exactly what it was supposed to do. It did exactly what the natural enzyme did, except it works only with the mirror image feedstocks that you have to feed it in. So you can't feed it normal ATP. It has to be the mirror image of ATP, which from a molecular standpoint is really hard to do. Like if you gave someone 1,000 right hands, you can't make a left hand out of that. You got to make its own left hand. Chemists and biologists made those and showed that life still worked when it was in this kind of mirror image universe in the lab. And so that strikes, so they've made things like that. In terms of where they are in terms of actually making a cell from scratch, I'm not quite sure. But it is an active area, and it is an area that's considered in this field of origin life research too. Yeah. Question?
I just have a question about your sodium potassium.
Yes.
Sodium and potassium are both alkaline metals in group 1, and they both have that same number of valence.
Uh-huh.
Why did you have to do all those experiments to predict what it would do in terms of the water turning? Why didn't they act the same?
Right. That's a good question. So sodium and potassium are right under each other on the very leftmost column of the periodic table, and usually things that are in the periodic table in the same column have do the exact same chemical reactions. They might be tuned a little bit differently, like carbon and silicon are, and sodium and potassium are kind of like that. They are different sizes, so that's going to have an effect of how water solvates them and everything. But we expected them to behave roughly similarly, and they do to a certain extent. But I guess what I'd say is they do behave similarly in the fact that they don't. Most of the time, they don't do anything interesting. But they just don't do what carbon does. They don't do what nitrogen does. They sit there in their countercharges. But there's going to be a point where some things are binary. Like this deliquescence is binary. You're either deliquescent or you're not. And you're going to be on some spectrum. And so if we were, like when I put up that tray, if we had pumped up the humidity much higher, every well in that tray would have been deliquescent. And if we pumped it all the way down, or down farther, everything would have not been deliquescent. It would have stayed dry. But there's going to be some humidities in between where one is and the other's not, and that's kind of, where we are in those experiments. So it's not that sodium salts can't be deliquescent, it's just that the humidities where they are much higher than they are for potassium. So there's something that kind of, the length of those bonds is longer, and thermodynamically how the ions are hydrated, it all adds up to potassium in this range as deliquescent, sodium's not. But in general, they do the same chemistry. Yeah. Other questions? It's almost 8:30. Question?
You're obviously a scientist working on science all the time. How do you respond or deal with what certainly feels like a very anti-science environment that
Right.
we're in today?
Yeah, so in terms of, I think that it didn't always used to be this way with science. When I was growing up, both sides of the aisle supported science and recognized its importance for our economy, for our national security. And I was on a scholarship in college called the Barry Goldwater Scholarship. Barry Goldwater, parents laugh because Barry Goldwater was one of the most conservative people in the Senate. And that support of science on that side of the aisle, you might imagine, is decreasing, but I don't think by all. I think that, but one aspect of the problem is, the way that I deal with this is that some people, I think, put their heads down and say, just get the job done. What you're doing, what you're thinking is important. But scientists need to do a better job of getting out there and talking to the general public and convincing people that what they're doing is important. Like, this is, of any area of science, a very emotionally, politically, theologically charged area, right? When I was at Harvard, our Origin Life Institute just started. At the first meeting, when we walked in, they posted armed guards at the door, police officers, because they did not know what to expect, like who would come in if they found it. And they didn't publicize the fact that we were having this kickoff meeting. Nothing happened, no one came. And later on, it was just the scientists that cared about it. But there was that worry, it was interesting. But I have never experienced, aside from an occasional weird email, I've never experienced anyone kind of, anything that was charged or threatening kind of there. But the answer to that question is that we have to do a better job as scientists about convincing Congress, the public, and ourselves. Scientists are pretty good about interacting with each other. They're not so good about interacting with the public. We need to do a better job of that and showing why these things are important. And, it's interesting, the Origin Life stuff is interesting, but just think, if we figure out what life is about, if we can come up with that, figuring out that water was H2O, what is that thing that makes life life? And what are those fundamental things that life relies on? There are going to be all sorts of discoveries that cascade out of that for medicine. We don't know why life insists on enriching potassium. There are diseases that are associated with potassium deficiencies or excess potassium. Maybe that will open up ways to treat those diseases. We just don't know until we do that basic research, and that's why this is so important from a medical technology standpoint, from an economic standpoint, because that's going to, as drugs are developed, as treatments are developed, there's a huge implication not only for health but for industry. And then there's just so much that can be done with technology that's developed in this area. So you want to focus on hard problems and trying to aim high and then get things that are associated with that. But the answer is, you got to do both. Like, my life is associated with teaching. That's the next generation of doctors that are out there and scientists that are out there. That's about a third of what I do. A third of what I do is the research in the lab that's focused on these tiny pieces of the problem that you kind of pick out for yourself. Collectively, everyone's got a little different piece of the problem, and you try to make progress. But then that third thing, what you do with the other third of your time is interacting with the community, publishing your results, and doing things like this, talking to people, because if you don't do that, then you're in big trouble. And even sometimes when you do that, you get into trouble but it would be much worse if you didn't. NASA is really, really good about convincing people that what they're doing is important, and it is important. But you might look at NASA and say, well, you really haven't gone out of low Earth orbit for what, 40 years, 40 plus years. So, but they've been so good at publicizing and talking about what they've done and milking those accomplishments for 40 years that people are really excited about NASA, as they should be. I wish people were as excited about the little blips that I see on the chromatogram that we pump out on a daily basis, because of it's kind of the same deal. It's not everything you do in the lab is sexy, but over time you hope to have some accomplishments that have a lasting impact and contribute to that knowledge base. But yeah, it's a really important point, and it's a point that I think is missed on a lot of scientists who would prefer not to talk to people, and would just kind of just want to stay in the lab and do their experiments. The experiments are important, but telling people about them is as important.
Just a comment, the American Association for the Advancement of Science has just formed the equivalent of a political action committee,
That is good.
which is about time.
That's good. Yeah, I hope they get a lot of dark money pumped into that so we could rival everything else.
You mentioned government putting their nose into science and the big global warming thing.
That thing, yeah.
That thing that some people don't believe in. And I was surprised that, you should ride your bike rather than drive a truck, because you're polluting the air and that's going to cause all kinds of problems and going to put us into another ice age. A friend of mine who dabbled in science says, no, there weren't any trucks around when the ice killed the dinosaurs.
Yeah, I mean, the Earth does go through cycles of this, and that's true, but yeah, the data suggests that humans are the ones who are responsible for it this time. And it is...
Nothing else is possible less.
Yeah, but I mean, we're in a world of alternative facts now, and just a propaganda machine. There are just instruments that people can use to get alternate facts out there, and it's an issue that that science is going to have to come to terms with. I think that scientists, for their part, are pretty good at distinguishing what's real from the noise. The problem is that if the people, a democracy relies on an informed electorate because of power and money come from the people. So if what the scientists are finding doesn't accurately make it back to the people who are making decisions like we did on Tuesday, then we've got an issue. We've got a problem. So that's the importance of a free press, and it's freedom of information, and how information is communicated is extraordinarily important. And it's there are no easy solutions here. There's a lot of noise. So
Separating fact
yeah.
from fiction.
Yeah, so you could just plug away and try to talk to people, and I think that a lot of people I think that most of the feeling even from people who don't believe in climate change, or who think that the origin of life is not something that should be worked on, that comes from, those are genuine feelings that they have, that they're not trying to, there's no animus there. So I think that people should be respectful in the debate and try to plug away and keep on with facts and reason and logic to convince each other, as opposed to the yelling at each other that you tend to see on cable news and things like that. Last question.
You were talking about the information that came back from the Hubble.
From Kepler, yeah.
Oh, well, any of this information,
Uh-huh.
enjoyed by what they
No,
got.
all right, so the first thing that happened with Kepler, if I recall correctly, was that they kind of, is that Hubble? The answer is they knew that they were going to get some data on this, but I think that what they got exceeded their wildest expectations. I don't think that anyone expected to find that many exoplanets. I mean, literally hundreds. These things are light years away. You know, planets are so hard to detect that, you know, we had a hard time on Earth detecting Uranus and Neptune. Like, that took a long time because those things aren't, you know, they're illuminated, they're not luminous, they're hard to see. But we're detecting the equivalent planets like way, way away. So there was a lot left. This instrument was really good. There was a lot of things that we got out of it. But the nice thing is, I talked about how it died, but there's this James Webb telescope, which is the next generation, which is, it's been delayed and delayed and delayed. So it should, I think it was supposed to have gone up by now, but it's going up very soon. And the instrumentation on that is going to allow us to do so much more and continue this work that kind of just ended as Kepler died. So stay tuned. I think in 5 to 10 years, we're going to have like just the whole next level of information about these places.
Did Kepler outlive its own?
I think Kepler outlived, well, I think that was pretty much designed, it might have just exceeded what it was designed to do.
They
It didn't,
really know, right?
right, it didn't die out early. It's kind of like how the rovers on Mars, like, vastly exceeded what they were designed to do. And we're getting so much good data about potential for life on Mars, or whether life exists on Mars and now isn't there. But NASA's incredible. How could you not be excited about all the stuff that they do all the time? And they are one of the major funders for origin life research because they want to know not just how life formed on our Earth, but how could life form on other planets? How do we detect it? And then if we detect it, like, what do we do with it? So there might be life as we don't know it out there, and it's just waiting for us to find it.